Hello, my name is Camryn Young. Today is July 10th, 2025, and this is my verbal presentation on the article Genetics-Based Methods for Agricultural Insect Pest Management. So pest management has become a very big deal in recent years, especially with the resistance built up to pesticides. One example of this is the diamondback moth, which has caused $4 to $5 billion of damage each year globally because it's built such a resistance to synthetic pesticides and the Bt toxin, which we'll talk a little bit more about later. But it kind of shows that this is something that can't kind of be brushed under the rug. It has to be dealt with pretty soon. So some techniques we've come up with is the sterile insect technique, or the SIT. This is a technique where a species that we want to have population control for is grown within the lab, and while they're grown in the lab, they are introduced to radiation to make them infertile. Once they are infertile and have reached maturity for reproduction, they are sent out to the geographical area where we want that population control, and they mate with the wild type species there, where the offspring will then be infertile. This has to be done over a long period of time, but is extremely beneficial in reducing populations. It's ideal that it's a biological and not a chemical intervention, so it's based off of the mating and not the chemical destruction of the geographical area. And it's also very cost effective. The costs isn't very high when comparison to the benefits that this creates, especially with it being so species specific. You don't really see a lot of off-target effects. We have used this in real life with the screwworm, and it has shown very positive effects. But we wanted to go a little bit further into it, which is when we added genetics into this. We noticed that it's most ideal when we send out simply male insects of that species because we want them mating with the female wild types. So we have created a female lethality gene where all the male insects of a lab-grown population will be homozygous dominant for this gene. So when they are released into the wild, only the males, because the females have died before they reached reproductive maturity. So when they all are released into the wild and mate with the wild type females, all the offspring, if they are female, will die before they reach a reproductive maturity, and all the males will reach reproductive maturity, but they will still carry this female lethality gene. So it goes further and further down each generation and maintains that population control. There are also incompatible insect techniques that have been seen to be working in mosquitoes, where again, it's only male mosquitoes that are grown and released, but they are given an intracranial bacterium that makes their offspring infertile. They are mated with the wild type females there, and population control is maintained through the sterile eggs that are kind of unable to produce offspring. Again, it's ideal that it's males and not females because you don't want the females to have this bacterium and then create a resistance to this. A lot of models have shown that this is effective in population control, but works best when it is combined with other methods. That other method we really like to combine it with is that Bt toxin I mentioned earlier. So we have our crops genetically modified to release a toxin from the, correct me if I misspell this or mispronounce this, Bacillus thuringiensis bacterium, which is like a natural insecticide. It's introduced at slow rates to create or prevent increased resistance through other insects for this, but something we decided that we could go further with because resistance is a recessive trait that is built up over time. While we release these lab-grown males with this homozygous dominant female lethality gene, we can also create a homozygous dominant susceptibility gene for Bt toxin and that Bt crop. So we release both of them, and so when this lab-grown insect goes out and mates with a wild-type female and they have offspring, not only do the females not reach the reproductive maturity, but the males contain a susceptibility gene for the Bt toxin. I think this is a really great idea overall with maintaining pest control and not creating extreme damage to our environment, as long as it is done safely and efficiently to prevent further resistance buildup.